Well, welcome back, everybody, to the last presentation, the last chapter of the semester, and also the longest and most complex chapter, because here we are delving into the chordates very briefly at the beginning, and then focusing most of our attention on a subphylum within the phylum Chordata, the subphylum Vertebrata, which of course is our own. Just remind you that while we seem to feel that vertebrates are the most important part of the animal kingdom, it really constitutes less than 5% of the overall animal kingdom. Remember, again, as we discovered earlier in a previous presentation, 95% or more of the animal kingdom is actually invertebrate. So we are discussing in this chapter just a small fraction, comparatively speaking, of the animal kingdom, but perhaps the part of the animal kingdom that we are most familiar with. So having put vertebrates in context, let's take a look at Chordata as a phylum. Then we'll talk about the two invertebrate subphyla of the Chordata phylum, and then we'll spend the rest of the time focused on vertebrates, the subphylum Vertebrata. So one of the things that we have to recognize is that for the longest period of time that life has existed on Earth, there were no animals with backbones. There were no vertebrates or even chordates. We find the earliest evidence of chordates, and therefore ancestors of all vertebrates as well, dating back to around the beginning of the Cambrian. Prior to that, any evidence of any sort of supporting tissue or any sort of supportive apparatus within the body of the very, very earliest of the animals coming out of the Ediacaran is essentially non-existent. So it is the Cambrian, and specifically the Cambrian explosion, that provides the first evidence of a new body plan. A body plan that is a literal 180-degree flip of the invertebrate body plan. Now, the reason why I say that is because you may recall, as we discussed the invertebrates in the previous presentation, that I made a point of pointing out that the nerve cord of invertebrates was always ventral. And what we find now is that in chordates and vertebrates, the nerve cord is dorsal. And what is interesting to note as part of this observation of this reorientation of the body plan is that the Hox genes that are involved in establishing dorsal ventral in invertebrates are the same Hox genes that establish dorsal ventral in chordates and vertebrates. The big difference is that the roles have been flipped. So the Hox gene that establishes dorsal in invertebrates is the Hox gene that establishes ventral in vertebrates. And likewise, the Hox gene that establishes ventral in invertebrates is the Hox gene that establishes dorsal in vertebrates. So you really can say that the chordate and vertebrate body plan with the dorsal nerve cord is a 180-degree rotation along the longitudinal axis, or around the longitudinal axis of the invertebrate body plan, to reposition the nerve cord from the ventral side of the body to the dorsal side of the body, with commensurate rearrangement of other internal features of the body plan as well. So you may recall that the digestive tract of an invertebrate tends to be more dorsal, sitting above the nerve cord. Well, if you look at the location of our digestive tract, it tends to be more ventral, sitting below, if we were to lie with the ventral surface underneath us, lying below the nerve cord, which is the location in most vertebrates and most chordates. And so you really can think of the chordate body plan, the vertebrate body plan, as a 180-degree rotation of the invertebrate body plan around the longitudinal axis to reposition the structures that were ventral to a dorsal location and vice versa. And this all seems to occur during this period of the Cambrian explosion. Some of the earliest evidence for chordates and perhaps even very, very primitive vertebrates are found in the fossil records that come out of the Cambrian explosion. And as you might recognize now, chordates and particularly vertebrates have come to dominate many of the ecosystems that we are familiar with, both or including marine, freshwater and terrestrial habitats. When we think in terms of the dominant predators and the dominant animal types, we tend to pay attention and notice the presence of the vertebrates as the dominant organisms in most habitats. And that's because having a vertebral column and having this supporting structure on which you can then hinge or you can hang appendages. In most animals, they are all legs, so the term tetrapod for 4 legs, but also the ability to modify those 4 appendages for other uses. So we know that in our case, with bipedalism standing on our 2 posterior appendages, that gives us the opportunity then to modify the anterior appendages, our arms, for other uses. And so primates, and that includes us, have these adaptations of the anterior appendages, what we refer to as arms in ourselves and in most primates, for other uses. And in humans, we see the greatest adaptation of the anterior appendages, the arms, for uses for other purposes. We'll get more into that when we start talking about primates and we address humans later in the presentation towards the end. So this is a fossil that comes out of China, if I remember rightly. This is Mylocunmingia. We'll talk a little bit more about this in another slide, but this is an early, we think, vertebrate. It does show evidence of a structural feature, let me fire up the pen, called a cranium. And so you can see that there is some sort of covering material that on the head. This is possibly bony material or maybe cartilage material, but it certainly forms a brain case and a covering over the brain. There is evidence of an eye. That's probably an eye located there. You can see evidence of segmentation. This is potentially a spinal cord or a spinal column that is a supportive structure within the body of the animal. At least this is how this is being interpreted. And this is a very, very ancient fossil. This goes back to this time period. Well, not exactly back to the Cambrian, but a little bit later, later than the Cambrian, a little bit closer to present. But it certainly shows evidence of structures that we would assign as typical of vertebrates, certainly of chordates, if not vertebrates. So when we start thinking in terms of vertebrates, we have to remember that vertebrates do not represent a phylum. They actually represent a subphylum of a larger phylum called Chordata. And within Chordata, there are in fact 3 subphyla, 2 of which are themselves invertebrates. And so the reality is that vertebrates, the animals that we pay greatest attention to, represent a small subsection of a much larger phylum. Being a chordate, they are part of a larger clade, the deuterostomia. We've talked about deuterostomes previously and what it means to be a deuterostome in terms of the developmental pathway. So if you're uncertain of what those characteristic developmental steps are, you might want to cast your attention back to either chapter 47, or you could take a look at chapter 32. Both the deuterostome developmental pathways are developed there. Of course, we do know that the name deuterostome refers to the fate of the blastopore, the fact that the blastopore becomes the anus and the second opening becomes the mouth, but there are other characteristics associated with deuterostome development that you also need to be aware of. That being said, when we look at the invertebrate clades, they are the urochordata and the cephalochordata, and we'll look at both of those clades or subphyla in a little bit more detail as we talk about the chordates as a general group. But the first thing that we need to do is identify what are the key characteristics of chordates, because there are in fact 4 characteristics that are present in all chordates at some point in their life cycle. And it's really important to stress that they are present at some point in their life cycle, because as you'll see as we work our way through this presentation, in some animals, many of the chordate characteristics disappear, including within ourselves. So we're now in your final adult form, as you'll soon recognize, we only retain one of the 4 characteristic chordate features, although we do possess all 4 of them during our embryological development. Some of them are present for a very, very short period of time, very transitory. In other cases, the features are co-opted and used to generate other structures that you'll become more familiar with as we continue on with this presentation. So what are the key characteristics of a chordate? Well, if we think in terms of, perhaps I should put them here because I want to talk about the phylogenetic tree a little bit later. If we think in terms of the key characteristics, you've got 2 of them listed here. So let me just put these in. Key chordate features. So one is the notochord. And we are going to talk about these in more detail a little bit later in the presentation. A hollow dorsal nerve cord, and remember we do have a way of explaining how this came about from invertebrates. Pharyngeal slits. So these are slits that are located in a region here on the neck, or what will become the neck in terrestrial tetrapods, but in fish give rise to the gills. And that is one of the first modifications of pharyngeal slits. Pharyngeal slits were actually used as feeding structures initially for filter feeding, and in some of the extant chordates still are, as you'll see, but they have been co-opted to give rise to other features. And when we talk about primates and we talk about specifically mammals, I'll show you how the pharyngeal slit structures have actually been co-opted to give us some very uniquely mammalian features. Pharyngeal slits and a post-anal tail. Now this last one is one that is often dropped, and people say there are 3 chordate features, but in fact the post-anal tail has historically also been included as a 4th common chordate feature. Because if you take a look at all invertebrates, the anus, exits the body at the very posterior end of the body. That is very typical of invertebrates. But in vertebrates, the anus typically is not located most posteriorly. There is an extension of the body. In many cases, it is supported by the vertebral column or an extension of the vertebral column, past the point of attachment for the posterior limbs. So in primates and in many tetrapod forms, it extends past the point at which the hind limbs attach to the pelvis or the pelvic bones. We actually do have a transitory postanal tail. It's present typically for a few days, very, very early on in our embryological development, but it typically is reabsorbed very rapidly. And there are very few traces of the postanal tail, although you do have some fused bones that make up what is referred to as the tailbone. It's actually, if I remember rightly, 3 or 5 small vertebrae at the posterior end of the vertebral column that are fused together to make up a single bone called the tailbone or the coccyx. And that is the vestige of your postanal tail. In the case of some individuals, there is an external evidence of that postanal tail, but typically in individuals that are born with that, it doesn't have anything more than cartilage. It doesn't have a bony support. It doesn't have musculature. It may have a few nerves associated with it. And typically it is surgically removed very early on because it is an inconvenience. It's not functional. It extends out of the body. It's a good thing to get caught on clothing and that sort of thing, and can get damaged. So surgically, it's typically removed. But for most of us, there is no vestige left behind other than the coccyx as the fusion of the bones that would potentially have formed the tail structure, and does form the tail structure in other primates that still have a postanal tail. So these are considered to be the 4 key chordate characteristics. We'll talk about them in a little bit more detail in an upcoming series of slides. In terms of the phylogeny of vertebrates as a group, the origin of the vertebral column is initially as a cartilaginous structure, not as a bony structure, but it is still considered to be a vertebral column, even though it's made out of cartilage. In the simplest of the vertebrates, or the most basal of the vertebrates, it is simply a cartilaginous supporting rod. It doesn't even encapsulate the nerve cord. In the lampreys, which are also considered to be basal, you do find that there are extensions from the cartilaginous column that do partially wrap around the nerve cord. So there is evidence of partial protection of the nerve cord. But we really don't see evidence of vertebrae that we would recognize them until we see calcification of the skeleton, which begins not with the skeleton itself, but in the jaws, and then proceeds to calcification externally before you see calcification of the skeleton by displacement of the cartilage. And you see that in all fish. So what is interesting is that there are a line of fish, we refer to them as cartilaginous fish, you know them as sharks and rays, that actually retain a cartilaginous skeleton. This is a juvenile characteristic. So this is an example of pedomorphosis, retention of a juvenile characteristic into adulthood and sexual maturity. And we know from the fossil record that sharks are descended from a bony ancestor. And in some sharks, there's still evidence of that bony skeleton or a vestige of the bony skeleton in the form of a smaller bony extension located primarily underneath the dorsal fin of some sharks. But when we start thinking in terms of a fully calcified or mineralized skeleton, we think in terms of bony fish. So we have several different types of bony fish. There are ray fin fish, which are the fish that you are most familiar with. These are the fish that we utilize as a source of food. But then there are two lines of fish that show evidence of the origin of fins as modifications of the fins, and these would be the lobed fin fish, of which there are two lines, the coelacanths and the lungfish. Lungfish are all found in the southern hemisphere, so unless you've been to South America, to Africa or Australia, or you've been to an aquarium that keeps lungfish, you probably have not seen them. But these are fish that do retain the ability to gulp air through an organ that is very similar to a lung and works very similar to a lung because it is vascularized. It can be used for gas exchange. And in fact, we know that this is a feature that has a very ancient origin because the earliest ancestors of all fish, whether they be the cartilaginous fish or bony fish, did have an air-breathing organ that functioned as a lung. And lungfish are descended from a line that retained that organ for air breathing. And in fact, even some ray fin fish have a similar sort of organ that allows them to gulp air in circumstances where the oxygen content of the water is insufficient for the gills to do gas exchange. In most fish, however, with the exception of the cartilaginous fish, that organ has been modified to something called the swim bladder that is used to maintain buoyancy. More of that when we talk about the fishes. So we now know that the tetrapod condition actually originates amongst the fish in the form of their pectoral fins, the fins that are located anteriorly, and the pelvic fins, the fins that are located posteriorly. That is the beginnings of the tetrapod condition. And this is reinforced by the fact that Hox genes that establish the pectoral and pelvic fins are active in terrestrial tetrapods, also guiding the development of the anterior and posterior limbs of all terrestrial tetrapods. So limbs originate not on dry land, but originate in water for purposes other than supporting the weight of the organism out of the water. They are convenient for scrambling around in the shallows amongst the litter dropped into the water by overhanging plants. This is taking place during the Devonian and on into the Carboniferous. When you see the appearance of the first forest, this is also the time period where you start to see tetrapods in the form of the earliest ancestors of amphibians making their first forays onto dry land, because now you have a food source in the form of arthropods, particularly insects, that are attractive, and the limbs that were convenient for scrambling around in the shallows turn out to be suitable for supporting the animal on dry land, supporting it against the force of gravity. So amphibians, or the ancestors of amphibians, make their appearance on dry land. Of course, they are still required to return to water for reproductive purposes, and their juvenile stages do their early development in the water. Then they almost all excuse me, undergo a form of metamorphosis to take on a form that allows them to live on dry land. But this tie back to water is an impediment to fully colonizing dry land. So the final adaptation is the appearance of the amniotic egg in reptiles and birds, and in early mammals, the monotremes, the egg-laying mammals. These are all in the form of shelled eggs, whether the shell be a leathery type of egg in the case of reptiles and the monotremes, or a calcium reinforced shell in the case of birds. It is a way of allowing the animals to reproduce on dry land without having to resort to laying eggs in water, because now the development of the embryos can take place inside a watertight container, the shelled egg, and the embryo can still develop in an quote unquote aquatic environment, the amniotic fluid. Mammals take that to or expand on that by, rather than laying eggs, in the case of the monotremes, the very, very earliest of mammals, they retain the egg within the body of the female. In the case of marsupials, early development takes place within the uterus of the female, but the young is born very, very early and very, very poorly developed. It makes its way into the marsupium, the pouch that the females possess, and there the young attach to a teat, a mammary gland, that allows them to then feed and complete their development inside the marsupium. In the case of eutherian mammals, all of the early development takes place within the uterus of the female. The support of the embryo, of course, is through the placenta. So an alternative name for eutherians is placental mammals. And the young are born capable of independent existence, independent of the mother. So the young are born very developed. However, depending on the type of mammal that we're talking about, this degree of development can be very highly developed. So something like a young horse is able to run with its mother within hours of birth. Or in the case of primates, the young are still very much dependent on their parents for long periods of time after birth. Again, depending on the primate, the length of time that the young spend with their parents and dependent on their parents for support varies. So that gives you a brief rundown of vertebrates, but we still haven't really addressed the chordates, those, and specifically the two invertebrate clades within chordata. So we need to go and take a look at those in order to understand the origin of vertebrates, because it's in looking at the two invertebrate chordates that we see the origin of vertebrates. So again, just to reinforce these shared characteristics, let's take a look at them again, just to remind you. So you have the notochord, which is a rod of supporting material. It can be cartilaginous in some cases. In other cases, it's made out of other material, but then does get displaced by cartilage to form a supporting rod. Dorsal to that, so closer to the back, is the hollow dorsal nerve cord. Remember, again, this is a relocation of the nerve cord from a ventral location to a dorsal location. You have the pharyngeal slits, which initially are for filtering food. So the earliest of the chordates are filter feeders using the pharyngeal slits for filtering food. But eventually these would be co-opted and adapted for gas exchange with the origin of jaws and devices for breaking food up by chewing. So the appearance of jaws and teeth would mean that you don't need to filter feed anymore. You can actually take larger chunks of food and break them up for the purposes of digestion. So the pharyngeal slits can be co-opted for other purposes. So they initially become gills. In non-aquatic tetrapods, they are co-opted for other features, and we'll get more into those as we progress through the presentation. Then you have the postanal tail that extends past the point at which the anus, the end of the digestive tract, exits the body or empties from the body. And so these are considered to be the four chordate characteristics, and there is a more detailed treatment of each of these on these next four slides, so you can go through these. And remember again that the PowerPoint is available on Canvas, so you can take a look at these. They're also well documented in the textbook. So one of the points of the nerve cord, and this is something that you see very particularly in vertebrates, of course, is that the nerve cord is going to give rise to the central nervous system. So at the anterior end, you're going to see the development of the brain. And again, it's in looking at the invertebrate clades of chordates that we start to see evidence of the origin of the brain. We'll talk again about that shortly. Then we've got the pharyngeal slits, and we've talked about the fact that these are initially for food filtering, and then they become part of a gas exchange apparatus, the gills. But eventually in terrestrial vertebrates, they contribute to the anterior end of the body by forming parts of the head and the neck, and in mammals, parts of the internal structure of the ear. More of that to come. And then the postanal tail. And remember again, many vertebrates do have the postanal tail. All chordates certainly have a postanal tail during their larval stage. In the case of one of the clades, the adult form does not. Many vertebrates have the postanal tail. We are somewhat unique in not having a postanal tail. So the two invertebrate clades of chordates are cephalochordata, common name the lancelets, and urochordata, common name tunicates. And when you look at the two, you might suspect that the lancelets, because of their body plan, are more closely related to vertebrates than the urochordata, the tunicates. And that for a long period of time was the dogma, that because of the body plan of cephalochordata, the lancelets, they were the ones that were most likely more closely related to vertebrates. The reality is, based on genetic sequences in recent times, is that the urochordata, in spite of what the adults look like, do actually represent the clade of invertebrate chordates most closely related to the vertebrates. So let's take a look at the two body plans, two groups of organisms, to better understand why this observation is negated by the genetic evidence. So here you can see the lancelet body plan, and it does retain all four chordate characteristics during the life of the animal. It is a very simplistic organism. Its body is maybe 1.5 to 2 inches long. If you actually want to see living lancelets, we are actually fortunate in that on the eastern side of Tampa Bay, south of the power station that sits in the northeastern corner of Tampa Bay, there are a couple of bayside parks that feature a sandy bottom in the bay when you wade out into the water. And if you go out and dig up some shovelfuls of sand out of there and run them through a fine sieve, screening material for a window or something like that would work very nicely. You can actually sieve out lancelets, because lancelets live in the sandy bottom of the eastern side of Tampa Bay. And if we were in the classroom, particularly if we were in the lab right now, I would show you a video that I have of these lancelets being extracted out of the floor of Tampa Bay so that you could actually see them and take a look at them. And in that video, they do actually point out the connection, genetic connection between the lancelet's nerve cord and the way it's arranged and our own developing nerve cord and the development of our brain. And we'll touch on that in another slide shortly. So these are the lancelets, cephalochordata. The pharyngeal slits are used for filter feeding. They do filter food particles out of the water, direct them into the mouth using these tentacles. The pharyngeal slits collect the food particles and pass them on into the digestive tract. The water then exits the body through this outflow called the atriopore. Its body does have evidence of segmentation in the form of these segmented muscles, and this allows it to swim in a very primitive fish-like manner. So if it is able to obtain all the food it needs in its current location, it will stay embedded in the floor of the ocean. But if the food stream peters out, it's able to extract itself out of the substratum, flick its tail from side to side. It's not a particularly smooth swimming action. It's very jerky, but it's able to flick its tail from side to side. And that allows it to move to a new location. It then inserts itself tail first back into the substratum and starts filtering food particles out of the water. So it's a fairly simple lifestyle, but it does illustrate the use of the 4 chordate features as part of the body plan. And you can see how it might be construed that this would be this body plan, because it represents a bilateral body plan very similar to our own, that these animals might represent, of the 2 invertebrate forms, the closest clade to our own. But that's not the case, as I pointed out. It is the Urochordata, or the tunicates, otherwise known as sea squirts, because of their ability to squirt large volumes of water out of their body. That represents the clade most closely related to vertebrates. And this is because as juveniles, their body plan is bilateral and it shows all 4 of the chordate characteristics. But the adults do not. And that's because tunicates undergo a massive reorganization of their body plan, a major metamorphosis that results in the loss of 3 of the 4 chordate characteristics. So here you can see the larval stage of a tunicate, and you can see that it possesses all 4 of the chordate characteristics. So it's got the pharyngeal slits that are used for food filtering. It's got the notochord, it's got the hollow dorsal nerve cord, it's got a postanal tail that extends past the point at which the anus exits from the body. It has all of those 4 chordate characteristics, and it looks very much like a bilateral animal. But shortly after the larvae develops, in the case of some, I mean, the larval stage in some tunicates lasts for minutes, literally minutes. In other cases, they may last for a little bit longer, hours, but eventually the larval stage settles down on the ocean floor and it attaches to the substratum via this region that anchors it to the substratum, and then it undergoes a massive reorganization, as I said, that results in an adult form that looks like this. So the water flows in, bringing with it food particles. They get filtered through the pharyngeal slits, the only chordate characteristics that is retained. The food then enters the digestive tract and gets processed. Waste is released through the anus, which exit into the excurrent, which is where the water exits the body of the animal. And they are able to maintain a fairly substantial water flow. And if they're surprised, they're actually able to forcefully eject large volumes of water through the excurrent. So this is where they get their name sea squirt from, because of this ability to squirt a substantial amount of water out. And they're referred to as tunicates because they have this structural feature that lies outside of the body, referred to as a tunic, that completely surrounds the adult form, not found in the juvenile form, the larval form, but certainly found in the adult form. And so when you look at these two body plans and you compare them to the vertebrate body plan, it's not surprising that people thought it was the lancelets that were the clade most closely related to vertebrates on the simple basis of morphology. But when you start thinking phylogenetically and you start looking at gene sequences, it becomes very obvious that the tunicates are the ones that are more closely related. So how do we explain this? How do we explain the origin of the vertebrate body plan from an animal that undergoes this major metamorphosis to an adult form that looks nothing like a bilateral? And the answer is pedogenesis. So remember, we have these two terms, pedomorphosis and pedogenesis. Pedomorphosis refers to the retention of juvenile characteristics to adulthood and sexual maturity. Pedogenesis is the process by which this takes place. And so if you have a tunicate larval form that simply does not undergo the metamorphosis to the adult form, but retains the forecaudate characteristics into sexual maturity, you can hypothesize or postulate the origin of a vertebrate body plan from a ancestral tunicate-like larval form, simply by the retention of the juvenile characteristics, the forecaudate characteristics of the larval form into adult form and sexual maturity. And so while there may be a disconnect between the adult tunicate body plan and vertebrates, the larval form certainly provides us with the evidence that we need to postulate the origin of vertebrates from a tunicate-like common ancestor, or a common ancestor with tunicates, by retention of the larval form into adulthood, rather than undergoing this metamorphosis. So if we think in terms of the origin of the vertebrate form, even though we do know that tunicates are the most closely related to vertebrates, we can still look at lancelets to get some clues as to the origin of the vertebrate form, because the Hox genes that are at work in establishing the body plan of lancelets, particularly of the very simple nerve cord system that lancelets possess in their adult form, something that the tunicates don't have, gives us some clues as to the origin of the vertebrate central nervous system. So when we look at the lancelet, the Hox genes that lay out the basic nerve cord of the lancelet, and it would be incorrect to refer to this enlarged swelling at the anterior end of the nerve cord of a lancelet as being a brain, because it's not. But its origin or its derivation is through the action of these two Hox genes, BF1 and OTX. Then the rest of the central nervous system, the posterior end of the central nervous system within the lancelet, is a product of this Hox gene referred to as HOX3. When we take a look at simple vertebrate embryos, we see the same three Hox genes in action, giving rise to what many neurologists refer to as the three sections of the brain: the forebrain, the midbrain, and the hindbrain. There is some controversy about whether we really should divide the brain up into these three sections. because the implication is that these three sections of the brain carry out different functions. And what we realize now is that the whole brain is involved in running our bodies. It's not that certain functions are strictly associated with specific parts of the brain. A more modern view of the brain suggests that the whole brain is involved in regulating activities, not just certain sections associated with certain activities. Yeah, I don't want to go into it more deeply than that. I've been reading some articles and a couple of books on this new thinking, and there's a lot of information that you can out there that's available for non-scientists, for lay people, that makes it fairly easy to come to terms with this new idea about how the brain functions. And if you're interested, that would be something to look at. If you're interested in the origin of the human brain, the mammalian brain, animal brains in general, there's a lot of interesting material that's being published, both in journals and also in book form, that is easily accessible. It's not written in, for want of a better word, scientific language. It's accessible to non-scientists and non-neurologists and neuroscientists, if you're interested. So take a look at that if this is something that interests you. But the important point here is that the early development of vertebrate brains is under the control of the same three Hox genes that lay out the very primitive nerve cord structure that you see in lancelets. And so there would be a connection between the very, very earliest of chordates and the origin of vertebrates. It's the role of the Hox genes, that these highly conserved regulatory genes that lay out the body plan, that give rise to the body plan. It's the same set of Hox genes in the lancelet and in the vertebrate embryo that are laying out the basic morphology of the central nervous system, or the nerve cord in the case of the lancelet. So even though lancelets are not the clade most closely related to vertebrates, they still give us some clues about the origin of vertebrates. So those are the chordates. Those are the chordate clades within phylum Chordata. From this point onwards, we will be starting to talk about vertebrates. But before we go there, let's take a quick break and take a look at some concept check questions. So I will be taking formal breaks throughout this presentation because it is quite long, and that will give you a chance to step away for a short period of time and be able to come back and pick up the presentation when you're ready at the point that you stopped. So here are the concept check questions for this break. Identify the 4 derived traits that are found in all chordates at some point in their life. And then you're a chordate because as a vertebrate, you are part of the phylum Chordata, but you lack most of the derived traits of chordates. In fact, you lack 3 of the 4 derived traits of chordates as an adult. Explain why this is so. I'll see you on the other side of the break with the answers.